Hello friends, welcome to BMH Learning. In this video, I'm going to talk about rocket immunoelectrophoresis. Rocket immunoelectrophoresis is a quantitative one-dimensional and single electro-immunodiffusion technique. One-dimensional and single electro-immunodiffusion means you will notice the movement in single direction as opposed to the counter-current electrophoresis. Rocket immunoelectrophoresis is an adaptation of radial immunodiffusion in which you also observe movement in single direction. Now, why we call this technique as rocket immunoelectrophoresis? It's because the plasprine bands appear in the shape of cone-like structures at the end of the reaction. Or you can say the bands are of rocket appearance. So what happens in rocket immunoelectrophoresis? In this method, antigen migrates in an electric field in a layer of agarose that contain an appropriate antibody. Now let's talk about the principle of rocket immunoelectrophoresis. The principle is such that in this method, the antibody is incorporated in the gel at a pH value at which the antibodies remain essentially immobile. So that means the antibody will not move. Under the influence of the electric field, the negatively charged antigens start migrating in the agar. As the antigen moves out of the well and enters the agarogel, it combines with the antibody to form immune complex which becomes visible. Now let's know how it happens. First thing we have to do is to prepare about 15 ml of 1% agarogel. The solution is cooled to 60 degrees Celsius and then antiserum is added into the agarose solution. It is well mixed for uniform distribution of antibodies. Agarose solution containing the antiserum is poured onto the glass plate placed on a horizontal surface. After the gel is uniformly spreaded, it is allowed to set for 30 minutes. The wells are made with the help of a gel puncher. 10 microliter of the stranded antigen with varying concentrations and test antigen samples are added to the wells. The setup is taken into the electrophoresis tank. 1x TBE buffer is poured into the electrophoresis tank such that it just covers the gel. The power cord is connected to the electrophoretic power supply according to the convention. Electrophoresis is carried out until the antigen travels 3 to 4 centimeters from the well. The glass plate is incubated in a moist chamber overnight at 37 degrees Celsius. Now let's talk about the result interpretation. The rocket peaks would look like this. A precipitation rocket spreading out from the loading well indicate positive reaction or specific antigen-antibody reaction due to the presence of antibodies specific to the antigen. The absence of the precipitation indicates no reaction, or you can say no antigen in the test sample. There is something important about the height of the rocket peak and its area. Both height and the area of the rocket peak are directly proportional to the amount of antigen in the sample. That is, the height of the plasprine peak depends on the concentration of antigens loaded in the corresponding wells. We can say more is the concentration of the antigen in the sample, more it would travel further in the gel. This was all. Thanks for watching.